Assalamu alaikum, dear students. I'm your teacher, Dr. Moinuddin, and I will teach you this course ABG 202 that primarily covers the basic concepts of genetics. So these lectures can be equally useful for the students other than the DVM classes, such as animal science, agriculture, or other related disciplines. This course, Animal Breeding and Genetics 1, is the first course of the DVM scheme of study that is currently being taught all across the country. The credit hours are 212, and the handouts can be received from the Google Classroom, which have already been shared. Uh, the couple of books which are very important for this course, and the chapters of these books are shared and will be shared with the students in future for the as a teaching material. The first one is Concepts of Genetics, that is written by Clark et al. 2018, and the second one is the a conceptual approach, Genetics, a conceptual approach, 2005, and written by Benjamin A. Pierce. Uh, as far as course contents are concerned, these are the course contents on your screen. Uh, the genetics, its historical development and scope, then the genetic basis of inheritance, gametogenesis, Mendel, its laws, application, and we try to make understood through the different examples, probability, uh, its concepts, laws, and its application, and application of the chi-square test to calculate the probabilities in genetic crosses. Then the modified segregation ratios, multiple allelomorphism, polygenetic gene inheritance, genes and different gene interactions that include dominance, recessive epistasis, additive, non-additive gene actions, genetics of the sex, that is the sex determinant mechanism, sex linkage and its variation, pleiotropy, linkage and crossing over, gene mutation, chromosomal aberration, lethal and sublethal genetic abnormalities, and finally the genetic engineering basic concepts is application through recombinant DNA technology, gene cloning and manipulation, and feature of this technology, and the last content is the extra chromosomal, extra nuclear inheritance. As far as practical is concerned, we will try to cover microscopic studies on the animal cells undergoing mitosis, meiosis, exercises on Mendelian and modified segregation ratios, karyotyping and bending for the chromosomal studies, then the genomic DNA isolation, purification, separation on gel, quantification of and storage, primers, how we can prepare them, polymorphism, chain reaction, PCR, DNA fingerprinting by using striction fragment length polymorphism, rapid AFLP, etc. And finally, the DNA sequencing and genotyping, and the result of the sequencing, how we can align them. So these are the contents which we try to cover during this course. And please wait for the next lecture. That will be the first lecture of this class. Thank you. Have a good time.